Richard K. Bruick, medicine. Anyone who has ever fallen into a swimming pool knows how quickly the body senses a lack of oxygen. Individual cells react much the same way. At UT Southwestern Medical Center, Dr. Richard Bruick has focused a great deal of his research over the past 10 years looking for the triggers that activate when cells are starved for oxygen.
We've been able to identify some key proteins that act as sensors within the cell that allow cells in our body to recognize what's going on in their environment.
The trouble begins when these sensors have a breakdown in communication. This failure to process information can lead to disease.
If cells that are involved in sensing iron and oxygen become damaged, for example, that can change the body's ability to respond.
Understanding how these proteins work could contribute to a number of medical treatments.
This is being pursued now by a number of pharmaceutical companies for treating anemia that is observed in patients with, for example, chronic kidney disease.
Richard's success at finding the proper pathway to help in the treatment of anemia led him and his collaborator, Dr. Kevin Gardner, to focus on finding a druggable protein to aid in the treatment of cancer, another disease characterized by breakdowns in oxygen sensing. This turned out to be a much bigger challenge.
The initial screens that were done to try to find drugs by many individuals in the field were unsuccessful. And what Dr. Bruick did was to come up with a new approach to screen for drugs.
At the time, I think it was kind of a crazy idea with respect to how most of the field was viewing this pathway, and we thought we saw an opportunity that we could exploit.
He's not a guy who's going to give up, and it was exactly that stick-to-itness that he exploited to find a drug that in fact could hit that undruggable target.
Richard's tenacity has paid off. Thanks in part to his access to the university's high-throughput screening lab, he's identified an impressive number of drug targets and compounds that are all currently being tested in the hopes of treating kidney disease and kidney cancers.
Most scientists would consider themselves successful if they identified a single compound that might be useful in the clinic. In Dr. Bruick's case, he's identified 3.
We really have some hopes that what we're doing is going to be useful to cancer patients and patients with anemia, and it'll be exciting to see in what other disease contexts that perhaps some of the tools that we've built to manipulate these pathways will be useful.